the chemical reactions. There are many types of chemical compounds which are present inside the cells, and they will undergo two types of changes. One is called as a physical and other is called as a chemical, or we can say that physical changes and the chemical changes. So here the physical changes includes the change in the shape or structure of the chemical compound without altering the bonds, that is without breaking or without forming the new bonds. So this is one type of physical change. The other one is a change in the state of the molecule, that is the ice will be converted into the liquid form, that is solid state is converted into liquid state. So but there is no formation or there is no breaking down of the bonds. So that type of physical change will be possible in different types of chemical molecules. But the chemical change which includes either the formation of new bonds or the breaking of the old bonds. So when old bonds are broken, the new bonds may be formed. So in this process, one chemical compound is converted into other chemical compound. So this type of change where the bonds are broken or reformed or the newly formed, such type of changes are called as a chemical changes which are taking place in the chemical compounds. And here the rate at which the chemical reaction is taking place or the reaction is taking place. So that is the rate of the reaction is shown by rate is equals to delta P by delta T, that is the number of products formed in unit time. So if the number of products are formed, it will be more in that particular unit time. So then we can say that the rate of reaction is faster. When the products that have been formed will be less, then we call it as a slower reaction. Likewise, the rate of reaction depends upon the number of products that are formed in particular unit time. Then here, as we are saying that the number of products will be more when the enzyme catalyzed reactions are taking place. But if the same reaction is taking place without catalyzing the enzymes, so that the products which are formed will be in that particular unit time will be less. And let us take the mechanism which is involved in the chemical reactions, that is enzyme catalyzed reactions. So but here before that we have to know something about zymogens or proenzymes. So these are the enzymes which have to undergo some sort of change before they participate in the catalytic reactions. So without undergoing such modifications, they can't participate in the reactions. Such type of enzymes are called as a zymogens or the proenzymes. So these are the one which will undergo some sort of modification or change prior to their catalytic property. So this type of enzymes are produced in the digestive process and also in the circulatory process, because in a digestive process there are some enzymes which have been formed which are called as zymogens. So if the zymogens are not formed or if they are directly participating in the catalytic property, so they start digesting the walls of that digestive system. So that is in the digestive process or in the digestive system. So these are useful to prevent autodigestion of walls of the digestive system. And in the circulatory system, these are useful to prevent the coagulation of blood inside the veins, or we can say intravenous coagulation can be prevented because of these zymogens. So likewise, some significances are related to these various types of zymogens, which are useful in the digestive process and also in the circulatory process of living organisms. So one example here, it is trypsinogen is an zymogen. Here it is converted into trypsin. So this trypsinogen is an inactive, or we can say that the proenzyme molecule. So that had to be converted into trypsin, then only they will participate in the process of digestion. So it is done by enteropeptidase. This is an activator. So this is a one which will help in the conversion of the trypsinogen into the trypsin molecule. Then only the trypsin can participate in the digestion of further biomolecules inside the digestive process.